Welcome to the MCAT Basics Podcast, brought to you by the physicians at Med School Coach. Each week, Alex Starks breaks down high-yield MCAT topics, so you can score as high as possible on test day.
Hello, and welcome to Prospective Doctor's MCAT Basics Podcast. My name is Alex Starks, and I'm really excited about today. We are going to start an entire series on metabolism. It's probably going to take us a couple months to get through, but it's so high yield for the MCAT, I think that sort of focus is warranted. So today we're going to talk a little bit about how metabolism is tested on the MCAT. We're going to go through glycolysis, and I'm going to give you my recommendation on how to study for glycolysis in particular, but it really does fit in for metabolism overall. All right, so metabolism, the big picture, it's all about energy. Well, duh. But when I say that, what I want you to imagine and to think about, and this is the lens that I really encourage students and test takers to view metabolism through, is electrons. So during catabolism, the breakdown of molecules, all of our cells, really all they're doing is taking away electrons from molecules like glucose and fatty acids through redox reactions, and then most of the time they end up in the electron transport chain. We're not going to talk about the electron transport chain, which I will call ETC from now on, in detail, but today we'll get there. But I'll keep referencing it because it does place a lot of useful emphasis on the point of cellular metabolism, which is make ATP so the cell can do whatever it needs. All right, since we're starting with carbohydrate metabolism, we're going to start with an overview of how glucose gets into your cells in the first place. So imagine you're at your favorite restaurant. They bring you a basket of bread to dive in. So as you're eating, chemical digestion begins with the secretion of salivary amylase. Now, salivary amylase is an enzyme which hydrolyzes long-chain polysaccharides into di and trisaccharide, but not cellulose, the fiber and stuff like that. And we'll talk about that kind of towards the end, like why. Now, as you continue to eat the bread, you might notice that it's tasting just a little sweet. Not like sugar, not like glucose, or fructose, or sucrose for that matter, but it does have a sweet flavor to it. And that's because those small saccharides, like the di and trisaccharides produced from the hydrolysis of that starch, can actually land on the receptors on the taste buds, in that as long as a receptor is activated, like we talked in biosignaling, that will send a message into the brain. All right, we're going to fast forward to the small intestine because nothing really interesting happens in the stomach with carbohydrate. And we're specifically going to talk about the duodenum. So here, brush border disaccharidases, always have a hard time with that word, further metabolize those di and trisaccharides into glucose. And then that's absorbed into the bloodstream. Now, this next point is surprisingly high yield and a fact you want to keep in mind. Once the glucose is in the bloodstream, the first place it goes is the liver, not the large intestine, not the heart. It goes into the liver through the hepatic portal. You know, as a side note, the liver is going to get a lot of specific attention, and I'm going to talk about it in particular detail throughout this whole series because it does have very distinct and important jobs. Just keep that in mind. All right, so the liver expresses a glucose transporter, and the abbreviation that I'll have for it is GLUT2, a glucose transporter class 2. So other organs, notably the pancreas, also express that type of transporter, the GLUT2. And so this transporter moves glucose into the cell from the bloodstream if the concentration of glucose in the bloodstream is above a certain threshold. And this is because the transporter has a dissociation constant, or KD, that requires a minimum concentration for really any meaningful binding and transport of glucose. So keep in mind, GLUT2 is based on concentrations. That'll come up later. All right. So because we just ate, our blood has plenty of glucose, and some of this glucose is transported into the liver, yada yada. But now the blood circulates the glucose through the body, allowing many organs, including the brain, in particular the brain, to use glucose transporters of their own to bring glucose in to those cells from the blood. Now, skeletal muscle and fat cells are unable to pick up glucose from the bloodstream at this point, or based on concentration by itself. They actually need a little bit of help. You may or may not have been wondering when I was going to get to insulin, and that time has come. So For skeletal muscles and fat cells or adipocytes, those are the only cells that require insulin in order for glucose, to be specific to say glucose, to be taken up into the cell. It does encourage glucose uptake in other cells, not the brain. Actually, that's important. But it does so in a different mechanism, and we're going to dive into that right now. All right, so we'll back up just a second and talk with the pancreas. So I mentioned that the pancreas also has those GLUT2 transporters that the liver has, right? And it's the influx of glucose into the pancreas through those transporters. And of course, that only happens if concentration is high enough, and that triggers the release of insulin. It's kind of a cool concept, I think. All right, so once released, the insulin acts on a variety of tissues. And the term that we use or associate with insulin is insulin-dependent glucose transporters. And those transporters are GLUT4. And what do you know? It's skeletal muscles and adipocytes that express these transporters. And I think this makes sense because you wouldn't want your fat cells and all your muscles to take all the glucose or to steal the glucose before your brain gets what it needs. Which, again, brain is not dependent on insulin for glucose uptake. All right, so that's the physiology background and kind of the organism level view. And we're going to zoom in and go to the cellular level. And we're going to use the liver as our example, but I'll call out any differences in muscle cells or other tissues if it's important. All right, let's set the scene. Glucose molecules are rushing into a hepatocyte from the bloodstream. If I asked Mr. Le Chatelier what would happen next, he would say that glucose would build up inside of the cell until it reached an equilibrium with the bloodstream, and then it would remain in dynamic equilibrium with glucose flowing back and forth through the transporter. But this would not be ideal because we want energy inside of the cell, not so much in the bloodstream. Now, the reason why we're able to not establish this dynamic equilibrium and we're able to pull in much more glucose from the bloodstream into the cell is our friend glucokinase. So pretty much just the liver has this enzyme, and we'll talk about why whenever we get to gluconeogenesis. So being a kinase, glucokinase transfers a phosphate onto glucose, and that chemically modifies and changes glucose. So again, that equilibrium is not established, but there's more utility here. So because glucose transporters are highly specific to glucose, it won't bind glucose with the phosphate on it. So I like to think about it as glucokinase is essentially trapping the glucose inside of the cell. All right, so now we have glucose 6-phosphate. That phosphate is transferred to the 6th carbon of glucose, which I'll now call G6P. And G6P has multiple fates here. It's like the context we're talking about today is glycolysis, but G6P, depending on other dynamics, could go through glycogenesis, or the formation of glycogen, or the pentose phosphate pathway. And we'll get to those, but again, today's about glycolysis. I want to pause for just a moment and talk about how we're going to go through all of the nitty-gritty of glycolysis. So when I work with a student, I teach metabolism as applied general chemistry and organic chemistry. It might be cringing, but the reason why is that it's easier to remember the logic of the processes, allowing you to memorize less, though of course memorization is absolutely necessary. I also recommend learning metabolism as a story. And my story is how I think about it is it boils down to how glucose gets mugged and robbed of all of its possessions, by which I mean its electrons primarily. It does lose some carbon and oxygens at some point too, but it's primarily the electrons we want to focus on. All right, so we're going to go through all of the steps so that you can have that resource in audio format, which I think is very helpful. But I'll also point out that the content that you absolutely need to have perfect by test day as we go through. Now, I've determined those pieces that you have to absolutely have perfect in other areas that are not quite as important because of released AAMC practice material, as well as analyzing how the AAMC tests metabolism in general. So, for example, if the AAMC has a question about what can and can't be produced from glucose or to be turned into glucose, well, that would suggest that you should probably know the potential fates of other related molecules like acetyl-CoA or fatty acids, things like that. All right, so the victim of our story, glucose, is a 6-membered ring, 5 of which are carbon, 1 of which is oxygen, and there is an additional carbon group attached to the 5th carbon in our molecule. Kind of sits as a hat, if you will, on top of the glucose ring. And each carbon has a hydroxyl group attached to it. The chemical formula is C6H12O6. All right, now let's talk about the story of glycolysis, or how I think about it, the subjugation, torture, and robbing of electrons from glucose in order to power an insatiable power factory called the mitochondria. All right, so step 1 of glycolysis, you must know this one. Glucose, free glucose in the cell, becomes glucose 6-phosphate, or G6P. This phosphorylation is carried out by glucokinase, primarily the liver, or hexokinase, almost everything that's not the liver. So the kinase hydrolyzes ATP, which provides abundant free energy, in order to catalyze the non-spontaneous transfer of phosphate to glucose. Carbon 6 of glucose has a hydroxyl substituent, and the hydrogen of the hydroxyl group is replaced by the phosphate group. So really just doing a swap. And as an orgo connection, this is really a fairly standard nucleophilic attack on the partially positive phosphorus in phosphate by the oxygen of glucose. It occurs at a relatively unencumbered position, carbon 6, so the steric hindrance is relatively small. All right, step 2. G6P becomes F6P. That'd be fructose 6-phosphate. So the cell prefers a different configuration of this monosaccharide, G6P, that it's stealing electrons from, and that's why it isomerizes it into fructose 6-phosphate. And this reaction depends almost entirely on the relative concentrations of each molecule, and so is readily reversible. The mechanism is pretty complicated and will not be pertinent to the MCAT, but structurally, all that has really changed is that the oxygen in the ring of G6P is now attached to carbon 2 instead of carbon 1. And that change is why fructose looks fairly different than glucose. All right, step 3. F6P becomes fructose 1,6-bisphosphate. Now, I can be in the habit of abbreviating this and calling it FBP, sometimes F16BP. So through this podcast, if you hear both or either or, just know I'm referring to this molecule. Earlier, we talked about how G6P can go to multiple pathways. So this phosphorylation step to make into F16BP is to ensure that glucose is committed to glycolysis. There's no turning back anymore. So if you know the structure of fructose, it should not be surprising that this phosphorylation event by phosphofructokinase 1 adds a phosphate group from ATP to the hydroxyl group on the first carbon. And the logic is the same as the phosphorylation we talked about of glucose at carbon 6. There's relatively negligible steric hindrance. Similar to step 1, it's a straightforward nucleophilic attack to replace the hydrogen on the hydroxyl of carbon 1 with a phosphate group. You must know this step because ATP is consumed and glucose is now committed to glycolysis. All right, step 4. F16BP is cleaved to produce glyceraldehyde 3-phosphate and dihydroxyacetone. And this seems like a relatively violent step in the process because after we've trapped glucose in the cell by phosphorylation, that poorer substrate is literally cut in half by the enzyme aldolase. Interestingly enough, it is completely reversible and does happen, and we'll talk about that in gluconeogenesis. These two molecules might seem abstract or hard to picture in your head, but I would encourage you to work on your chemical foundations so that you can deduce the structures if you're asked to. I'll do my best to describe them for you though. So glyceraldehyde 3-phosphate, or G3P, is 3 carbons. Carbon 1 is an aldehyde, glyceraldehyde. Carbon 2 has a hydroxyl substituent, and that's probably the more difficult one to remember. And carbon 3 contains a phosphate group. It's not so bad. Dihydroxyacetone phosphate is also 3 carbons, and the only difference is that instead of an aldehyde at carbon 1, we have a ketone at carbon 2. So the only difference, carbon 1 is a hydroxyl, carbon 2 is the carbonyl, but carbon 3 still has the phosphate group. In the mechanism, which is complicated, the C4-C3 bond of F16BP is cleaved in the reaction. Probably don't need to know that for the MCAT, But if you can remember the structure of F16BP, it may help you to make sense of what's occurring and help keep a track of where the carbons are going. This step is important, and the structure of G3P in particular is important because this is an area that connects to fatty acid metabolism. All right, step 5. So now the cell is going to take DHAP, or dihydroxyacetone phosphate, and isomerize it into G3P. This is catalyzed by an enzyme called triose phosphate isomerase. All right. So this step is completely practical on the part of the cell, because if you think about it, if it can turn DHAP into G3P, well, then there's just one pathway, one set of enzymes that's needed to further metabolize this molecule. All right, so again, this step exists so that the cell can be as efficient as possible in stripping away all of the electrons that these molecules hold dear. These last 5 steps are kind of like the setup phase, if you think about it. And we have gotten the molecule in the shape with the right functional groups, blah, 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 that the cell wants in order to move into what's called the payoff phase to strip away all those electrons. It's the oxidation of molecules that were once glucose. And now, of course, as we know from Gen Chem, oxidation of a molecule implies another molecule is reduced, i.e. gains electrons. This step involves the enzyme glyceraldehyde 3-phosphate dehydrogenase, and takes G3P, both of them. So now this is the point where we start multiplying things by 2. And it turns it into 1,3-bisphosphoglycerate. And in that reaction, we produce 1 NADH. And that is per molecule of G3P. So 1 molecule of glucose entering glycolysis, there are 2 NADH produced in this step. These electrons that are shuttled in the form of NADH have 2 ultimate fates, which we'll talk about after we get through all the steps. All right, so the mechanism here is not particularly important. Just note that the electrons in the CH bond of carbon 1 of G3P, I'm hoping you can start to visualize what this looks like, are stolen by NAD+, an inorganic phosphate is also added to the aldehyde oxygen of G3P. And so now we have 1,3-BPG. It's important to remember that this is a phosphorylation step that does not use ATP, actually. It uses a phosphate that is essentially freely floating in the cytosol, and we refer to that as an inorganic phosphate. All right, step 7. Like step 6, everything I described will be occurring in double. So now we have 2 molecules of 1,3-BPG. So this molecule is acted on by phosphoglycerate kinase. The kinase actually transfers a phosphate group from 1,3-BPG to make ATP from ADP. The resulting molecule is 3-phosphoglycerate. Now, something that I've always found odd was that we go from 3-phosphoglyceraldehyde from step 5, we add a phosphate step 6, and then we just turn around to remove it in step 7. But if you look at the molecules, they've been oxidized, and that aldehyde is now a carboxylate, hence the suffix -ate, which indicates it is the conjugate base of an ic acid. So this seemingly complicated turn of events fits our logic of metabolism, though. Steal all the electrons. Step 8 is a seemingly picky step. It involves phosphoglycerate mutase, and all that happens is that the phosphate group on carbon 3 of 3-phosphoglycerate is moved to carbon 2, and the hydroxyl group from carbon 2 is moved to carbon 3. This step does fit our theme of the cell wanting these molecules in a certain arrangement, and it has a dedicated step to make sure that happens. Step 9 is slightly more interesting, but does help to make sense of step 8. So the enzyme here is enolase, and it catalyzes the dehydration of the hydroxyl group on carbon 3. Apparently, it was unable to perform a dehydration reaction when the hydroxyl was on carbon 2, hence the need for that isomerization in step 8. Now, if you're following along with how the molecules are looking, this step creates a double bond between carbon 2 and 3 when the hydroxyl group leaves as water. During that formation of the double bond, the electrons associated there are transferred to NAD+, and we create NADH. The name of our product here is phosphoenol pyruvic acid, usually found as phosphoenol pyruvate, or PEP. Now keep in mind, the NADH that was formed, we have to multiply by 2. So now we've created 4 molecules of NADH. Step 10, the final step, sort of. Okay, so this step is really interesting and is also of utmost importance to know. So pyruvate kinase, the enzyme, does a reverse move, similar to step 7, and transfers the phosphate that is attached to carbon 2 in PEP to ADP and forms ATP. The molecule formed is pyruvate, and the structure of pyruvate I think is very important to know. And unfortunately, at least to me, it's not intuitive. But here we go. Here's the structure. 3 carbons. Carbon 1 is a carboxylate, so COO. Carbon 2 is a ketone, and carbon 3 is just carbon 3. So to recap glycolysis, the overall reaction of glycolysis is 1 glucose plus 2 NAD plus 2 ADP plus 2 inorganic phosphate yields 2 pyruvates, 2 NADH, 2 ATP, and 2 water. Steps 1 and 3, we invested 2 ATP. Steps 7 and 10, we got out 4 total ATP. So when the MCAT asks you how many ATP come from glycolysis, what they're really asking you are what is the net, or what we call the visible ATP produced, and that's going to be 2. I want to focus in on the 2 fates of pyruvate and NADH that were produced. In real life, there are multiple different things that can happen, like in different kinds of cells and whatnot, organisms, but for the MCAT, there are just 2 that you need to know. And they're a pair because where one goes, so does the other. And that fate is essentially determined by the presence or absence of oxygen. Again, it's a little more complicated, but for MCAT, stick with O2 or no O2. Now, condition 1, when there is no oxygen, the pyruvate and NADH stay in the cytosol and they undergo anaerobic respiration. It produces lactic acid and it regenerates NAD+, so that it can continue to participate in glycolysis. So with this step, NAD+ now can go back to where glycolysis is occurring and accept more electrons. So the lactic acid that you produce here is shipped over to the liver. Here it undergoes what's called the Cori cycle and turns it back into pyruvate. So there's no indication that the Cori cycle is required knowledge for the MCAT. So I think it suffices just to understand that lactate made in anaerobic respiration can become pyruvate again. Now, if there is oxygen in a nucleated cell, the pyruvate and NADH will be transported into the mitochondria. The NADH will drop off the stolen electrons to complex 1 of the electron transport chain, and the pyruvate will enter into the pyruvate dehydrogenase complex and be converted into acetyl-CoA. That acetyl-CoA will either be oxidized for its electrons through the Krebs cycle, or it can be turned into fatty acids. That's why around the holidays, when you eat too many carbs, you might get a little fluffy. This is also why drinking alcohol can quickly help you gain body fat, because alcohol is metabolized into acetyl-CoA, and if you have a bunch of acetyl-CoA floating around, it's not much work for the cell to turn it into fat. I'm just going to emphasize one more time how I think you should study glycolysis and metabolism in general, and I think you will be rewarded by the MCAT if you do approach it like this. While approaching it, memorizing and also thinking about the principles, the chemical principles, and the logic of what's occurring. Memorizing helps you learn the foundations and like that logical, and vice versa. The logic and the chemical foundations will help the memorization process too. I've provided mostly information about the chemical principles and logic. For example, when we talked about why DHAP would be converted into G3P. So if that makes sense to you, this story of pillaging and robbing glucose of all these electrons in order to power the electron transport chain, if that makes sense, then really all you need to do now is to memorize and the repetition and have your spaced repetition and studying so that you don't forget it on test day. The types of questions and the forms of questions that the AAMC will throw at you on test day typically have to do with like cause and effect. Again, they could ask you some random nitty-gritty, but very straightforward question, but that's not really their MO. They like to make you think. So the question style would be, let's say we inhibit a particular enzyme in glycolysis. And what would happen to ADP or ATP or NAD plus or NADH or cellular or cytosolic glucose concentrations? So those are the types of thinking and mental gymnastics that you would need to perform. Alrighty, quiz time. And if you're not finding these quizzes helpful, or if you'd like to see them different, or you do like them, or you would want more of them, more questions at the end, just let me know. Question 1. What is the purpose of phosphorylating glucose? I'll give you just a second, but of course pause if you want longer. All right, so the answer is not to commit it to glycolysis. That's the second phosphorylation event. It's to commit it to a cellular metabolism. And that's my second question. Why is it now committed to cellular metabolism? It's because the phosphorylation event that occurs in that first step traps it inside of the cell because the glucose transporters we talked about can't move a phosphorylated monosaccharide. I just thought of a third question to piggyback that off of that would be very, very MCAT style, would be to think why it can't transport it. And the theme that you'll run into on those is going to be either the charge, because phosphate has 2 negative charges on it in the cell, or the shape of the molecule is going to prevent its movement. So the actual second question is why we can't digest cellulose, which is very, very similar to starch. It's a linear long chain polysaccharide. Give you a second to think about that. All right, so the answer is that the linkages between the monomers in cellulose are in the wrong conformation for our enzymes, which tend to be stereospecific. So starch has an alpha linkage between the glucose monomers, but cellulose has a beta linkage. And just think, if we could package that enzyme into something, like sprinkle on food, we could solve world hunger overnight. Tree bark would even start tasting good. And cows love grass because they're able to get energy from it. But something that you should maybe know is that it isn't because cows possess the proper enzyme for cellulose. It's because the bacteria in their gut do. So the bacteria use their enzymes, and the cows are able to metabolize the smaller saccharides that are produced. We actually get vitamin K, humans, and I think other animals, but we actually get vitamin K because bacteria in our gut do the same thing. This third one is much more AAMC style than the last two. And it's not something that we've talked about explicitly, but is very, very important to consider. And I would ask, why are electrons transferred from glycolytic intermediates to NAD+? We'll do multiple choice on this one. So A, NADH has a higher reduction potential than intermediates. B, NAD+ has a higher oxidation potential than intermediate. C, NADH has a lower reduction potential than NAD+. Or D, NAD+ has a higher reduction potential than intermediate. All right, let's think about what answer A would imply. So if NADH has a higher reduction potential than intermediates, well, that doesn't really make sense. NADH is the form that has the electrons, and I'm pretty sure you can't reduce NADH any further. All right, so for B, NAD+ has a higher oxidation potential than intermediates. This is pretty much the same law as answer choice A. NAD+ is the oxidized form. So saying it has a high reduction potential, or even considering its reduction potential, doesn't really make sense. Unless we knew, of course, that NAD+ could be further oxidized, then talking about its higher oxidation potential could make sense. All right, so for C, NADH has a lower reduction potential than NAD+. I mean, that's certainly true. NADH has the electrons, so it's not going to have a high reduction potential, and NAD plus wants electrons, it's positively charged, so it would have a higher reduction potential, but again, that answer choice doesn't really mean anything. That would be like trying to compare the reduction potential of calcium 2 plus and calcium. And for our correct answer, NAD plus has a higher reduction potential than intermediates. So the only reason, of course, that electrons flow from glycolytic intermediates to transport molecules is because the oxidized form, or like NAD plus, of the transport molecule is higher than the potential of the intermediate that is oxidized. So the concept fits in really well with our theme and discussion today about the importance of viewing metabolism through that lens of foundational chemical processes. Well, that does it for our glycolysis episode, our very first installment of many to come from Metabolism. I hope this was useful for everybody who is studying for their MCAT. And as always, if you have any questions, comments, concerns, anything like that, feel free to send me an email at alex@medschoolcoach.com.
Each episode of MCAT Basics is brought to you by Med School Coach. To access Med School Coach services, including MCAT tutoring and medical school admissions advising, visit our website at medschoolcoach.com. Good luck as you prepare for the MCAT, and we hope you tune in again next time.